Hello, this is my project. My final project is about AI in drug discovery. The motivation of the study is uh, based on the assessment of the number of molecules that there are in the number of potentially viable small molecules is over 10 to the 60. But testing them in real-world clinical trials is extremely time-consuming. And as a result, in last year, 2023, only 55 new drugs have been approved by the US Food and Drug Administration. To make things worse, over 30% of drugs have failed in human clinical trials because they are determined to be toxic, despite promising preclinical studies on animal models. So this represents a huge cost in human and financial resources. Therefore, a computational drug toxicity assessment to test whether certain chemicals have the potential to disrupt the process in the human body would be of great value to the drug development effort. Chemical data is recorded in different ways. The most common and popular way is the two-dimensional representation that shows the structure of the molecules in 2 dimensions. But for data processing, the easiest way is to use the SMILES notation. The SMILES notation is a 1D string. Basically, it's a string notation in 1D. So it's a sequential notation. For this study, now we are going to leverage data set from a vast Kaggle competition. The data set contains about 8,000 samples and 200 samples for validation. And then there is unlabeled test examples that need to be scored and submitted to Kaggle for computing the result. Now, the data set includes SMILE strings, and in that particular study, because it was for some course, apparently, they had pre-computed the one-hot encoding of the SMILES. But we do not want to use the provided one-hot features, and we prefer to develop a notebook and to recompute it ourselves. In the data set, we, as I said, we find 8,000 examples, and in those 1,000 examples, we are looking at the distribution of the alphabet, the characters in the SMILES alphabet. And we can see in this histogram, C for carbon is the most frequent element in those molecules, and is followed by the equal sign that means a covalent bond. We observe a difference of 5 orders of magnitude between the most frequent element and the least frequent element, which is off the chart, because if I put the whole chart, it wouldn't come out well in PowerPoint. Okay, but chemical elements can be one character, like these examples, like carbon and nitrogen, but they also can be 2 characters. And in order to do in order to tokenize the data set, we tend to use the periodic table that it will help us to recognize the 2 character strings And in that way, we can decide that certain pairs of characters, like this NA or BR, are um, are a token and are a separate token of themselves. In that case, we have repeated the histogram, and we see that the elements, which elements are the less common, and the less common elements have appear only once in the whole data set. Like we show an example down here. Scandium is a rare earth that it was included in one of the molecules and is considered non-toxic. So the next thing is to look, next we look at the SMILES length. The length of the SMILES strings vary greatly. This we can see that the complexity of the molecules in our data set varies from very short ones, like under 50 tokens long, to almost 400 tokens. We look here at the smallest of the molecules, and this one, and here you are looking at the representation of the molecule in 2D, and the most complex one is the next one, and you can appreciate the wide range of complexity from the simplest to the most complex. We also look at the class distribution between non-toxic and toxic molecules in the label data set. And we appreciate that we have a ratio from 7:1, and that means that we might want to use weights to compensate for this class imbalance when we train the classifier. We are going to encode we're going to encode the SMILES strings for our model to in the one-hot encoding notation. So for that, we will have to generate matrices, 2D matrices that will encode each of the SMILES strings. So this is an example of one SMILES string. The string is at the top. As you see, this is the description of the string here at the top. And what you see here at the border, on the left border, is the vocabulary of the SMILES notation. So each symbol will have a position in the vocabulary. And if we put those symbols along the y-axis, we will have one row for each one of these symbols. So as you can see, for the symbol carbon, you have to fill in these two cells because the string has carbon in columns 1 and 2. Okay, sorry. I didn't mean to switch to the next string, to the next slide, but it's okay because we'll continue talking about the same thing. What you see now is the image of a one-hot encoding matrix for a single example. And this is for the longest string. So the longest string is just below, and this one has close to 400 tokens. And if you look over here, you will see the Y points going all the way to almost 400. And again, in this image, the vertical lines are, each one of the vertical lines costs only one character, and the position of the y point in the vertical line represents the symbol contained in that column. All right. Well, we're moving to our results. We have trained 3 models, and here you can see the first one, how it responded to during training. The 1D convolutional model is the smallest model that we train, and we are appreciating that it does learn pretty well up until 300 or 400 epochs. By the time it's hovering around 70% validation accuracy, and then it starts to overfit. Now, this is probably because we don't have so much data for the complexity of this feature space. We only have 8,000 examples, and if we try to train a model of this size with 8,000 examples, it will probably will have to, at some point, it will start overfitting. Next, we try with a transformer encoder. So transformer encoders are used for to build classifiers, right? As opposed to the transformer decoders that are the most famous ones because they are used in generative AI. Now, we train a transformer encoder, and we have 2 variants of that. The first one that we're showing here, it's trained with 1-hot encoding inputs. Now, the same 1-hot encodings that we use for the previous classifier. And as you can see, it's much slower to learn. It has a lot more parameters, and it will probably continue learning, but it was interrupted by early stopping because the early stopping was associated with the loss. So even if you don't see the loss here, you're looking at the binary accuracy, and it seems to be progressing. The loss maybe wasn't progressing, and then after 500 epochs, it was interrupted. Now, nevertheless, it doesn't look like it's learning enough, and this model, when we submit it to Kaggle and look at the test accuracy, it was only 35%. Now, here's the results in Kaggle, right? We can see at the top the result from the transformer model, and at the bottom the result from the convolutional 1D model. And in this case, we are completely sure that the smallest model learns faster and better, and that is probably because but it ends up overfitting at the end. So that is a motivation to think that if we had a lot more data, we could train the transformer model that is more complex and runs slower for a lot longer, and it will probably overtake the performance eventually of the small model. This is all my presentation, and it has been just a small introduction to the field of drug discovery for me, and answer to identify a series of next steps. Now, here are the next steps that we have identified. We want to start leveraging BERT models that are pre-trained with SMILE strings, and they could be used to generate the features for a thin layer classifier. Now, as a more maybe ambitious, slightly more ambitious way of transfer learning that we could use is just fine-tune the BERT model. So we can use our label data set to train the same thin layer classifier at the top, plus 1 or 2 layers of the model. And we could also adapt our classifiers to accept pre-trained embeddings as input. And there are some people who have pre-trained embeddings with smiles. That would be an interesting thing to try. There is also the chance, as you can see, there are 2 representations, at least 2, and there are more actually, representations of chemical molecules, and we could actually train a multimodal classifier that accepts the SMILE strings plus the 2D structural images, and basically use the 2 things to classify the molecules.